It was the most ambitious marine restoration Scotland had ever attempted, and it began with just 300 oysters. In March 2017, a team of scientists carried them out into the Dornoch Firth and lowered them into water that had been without the species for over 100 years. 8,000 years of native oyster reef, gone in a single generation of industrial dredging. What those 300 oysters did to the water changed Scotland forever. On the morning of the first deployment, Professor Bill Sanderson of Heriot-Watt University watched his team lower the ballasted bags beneath the surface. The oysters settled into water that had not held their kind since the late 1800s. Sanderson had spent 3 years in archives before this moment, reading navigational charts, radiocarbon dating shell fragments, and building the scientific case that this Firth could sustain oyster reefs again. Now there was nothing left to do but wait and see if the water agreed. From the shore, the Dornoch Firth looks pristine. The most northerly estuary in the UK stretches wide and grey under highland skies, flanked by low hills and empty coastline. The kind of place that appears in tourism photographs as evidence that Scotland's seas remain untouched. A person standing on the bank would see nothing obviously wrong. That was the problem. The Firth looked healthy. It had looked this way for so long that almost nobody remembered what it had once contained. For more than 8,000 years, native European oysters, Ostrea edulis, covered the seabed of the Dornoch Firth in dense, living reefs. These were not individual oysters scattered across mud. They were structures, layered colonies of shell and animal that built complex habitat on the seafloor, rising from the sediment, creating surfaces for other species to colonize. The reefs altered water flow, concentrated nutrients, filtered the Firth continuously. They were the seabed's operating system. Then came the dredgers. In the late 19th century, industrial oyster fishing swept Scotland's firths with a thoroughness that left almost nothing behind. Overfishing reduced stocks faster than the oysters could reproduce. The last native oyster in the Dornoch Firth disappeared sometime around the turn of the century. The seabed didn't look different afterward. That was the trap. The water was still there. The Firth was still there. But the living architecture beneath the surface, the reefs that had structured the ecosystem for millennia, was gone. And without it, the Firth was quieter than anyone who stood on the shore could hear. Bill Sanderson had spent years studying what oyster reefs do to the water around them. As a marine ecologist at Heriot-Watt University in Edinburgh, he understood something that was not obvious from the surface. Oyster reefs are not passive features of a seabed, they are active systems. At reef scale, millions of animals across hectares of seabed, their filtration capacity is staggering. It shapes water chemistry. It drives biodiversity. It determines what can live in the Firth and what cannot. In 2014, Sanderson became the research lead for something that had not been attempted before in a marine protected area in Europe, restoring a native oyster reef from extinction. The project was called DEEP, the Dornoch Environmental Enhancement Project. Its partners were Heriot-Watt University, the Marine Conservation Society, and an unlikely third party, Glenmorangie Distillery, which had stood on the banks of the Dornoch Firth since 1843, and whose distillation process produced organic wastewater that discharged into the Firth. Glenmorangie's involvement was not purely altruistic. The distillery understood it had a problem. Its production process generated effluent, the organic byproduct of whiskey making, that entered the Firth under a regulated discharge license. A restored oyster reef offered something a pipe in a treatment tank could not, a living solution that would improve over time rather than simply manage the damage. The partnership was unusual. A whiskey company, a university, and a conservation charity, but the logic was more uncomfortable than it first appeared. For most of its 170 years on the Firth, Glenmorangie's relationship with the water was one-directional. Water came in and effluent went out. The company that had contributed to the Firth's degradation was now funding the science to reverse it. And the thing the distillery had been putting into the Firth, organic effluent rich in nutrients, turned out to be exactly what oysters eat. The waste that had degraded the Firth for generations would now feed the animals rebuilding it. The polluter had become the restorer. And it worked precisely because of that contradiction. Before a single oyster entered the Firth, Sanderson spent 3 years in the archives. Trawling navigational charts, fisheries records, museum collections, and ancient literature, he built the scientific case that reintroducing oysters was ecologically legitimate. Shell material was sampled and radiocarbon dated. The results showed oysters had been present in the Firth continuously from the end of the last glaciation until their extinction in the late 1800s. The next problem was the seabed itself. A century without oysters meant a century without the hard substrate that oyster larvae need to settle on. Native oyster spat will only attach to shell material. On a bare sediment bed, they drift and die. To recreate the conditions that once existed naturally, the team used waste shell from Scotland's scallop and mussel industry, laying it across sections of the Firth floor to form artificial substrate. Shell waste became the foundation for something that had once built itself. Then came the question nobody could answer from a desk. Would the oysters survive? Nobody had done this before. That was the problem nobody said aloud at the start. When deep began, there were almost no established protocols for restoring native oyster reefs in a marine-protected area in Europe. Sanderson's team was building the methodology as they went, deciding how to lay shell substrate, which densities to target, and what survival rates would constitute success or failure. Every decision was made without a template. As Sanderson later described it, they were trailblazing all the way. The licensing alone took years. Working in a special area of conservation meant every intervention required regulatory approval. Biosecurity protocols had to be established from scratch, ensuring oysters sourced from Loch Ryan carried no pathogens that could affect the Firth's existing marine life. The team varied shell type, shell density, and placement method across different sites, not because they knew which approach would work, but because they did not. Monitoring happened every 6 months. The gaps between dives were long. You put oysters into cold, dark water and waited. Sanderson called the first 300 oysters canaries in a cage. You put canaries in a cage to find out if the air was still breathable. You did not know the answer until you checked. 300 oysters sourced from Loch Ryan, the only remaining sizable wild native oyster population in Scotland, were placed in ballasted bags at 2 sites in the Dornoch Firth. It was a survivability trial. Nothing more. If the oysters died, the project would need to understand why. If they survived, the next phase could begin. Sanderson and his team monitored the sites over the following months. The oysters opened. They began filtering. They did not die. The survival rate reached 86% at one of the 2 sites, in line with the team's highest expectations. The chemistry was right. The temperature was right. The conditions that had sustained native oysters for 10,000 years were still there, waiting beneath a surface that looked no different from any other estuary on the Highland coast. The 300 became the foundation for something larger. What an oyster does to water is invisible from the surface. That is what makes it so easy to underestimate. Each oyster draws water in through its gills and passes it across a mucus-coated filter surface. Phytoplankton, bacteria, and suspended particles, everything small enough, is captured, concentrated, and deposited on the seabed as dense, nutritious organic material. The water that leaves the oyster is clearer than the water that entered. Do this across thousands of animals, across hectares of reef, continuously, and the effect accumulates in ways that reach far beyond the individual animal. Cleared water allows light to penetrate deeper. Deeper light supports more seagrass. More seagrass creates more habitat. The reef structure itself, layers of shell colonized by algae, worms, crustaceans, and small fish, becomes a biodiversity hotspot on a seabed that had been biologically impoverished for a century. Sanderson's team studied Scotland's last intact native oyster fishery at Loch Ryan to model what a restored Dornoch reef might produce. Their findings, published in the peer-reviewed journal PLOS I, predicted that biodiversity in the restored areas would double within a decade of reef establishment. Double. Not an incremental improvement. Double. And there was something else the research identified that nobody had initially set out to measure. Oyster shells are composed largely of calcium carbonate. As oysters grow, they lock carbon into their shells in a form that persists long after the animal dies. A reef of 4 million oysters covering 40 hectares of seabed represents a significant long-term carbon store, one that grows as the reef expands. The Dornoch Firth was becoming a model for what marine restoration could contribute to climate. By May 2024, 90,000 native oysters had been returned to the Dornoch Firth. The numbers had grown in stages: 300 in 2017, 20,000 by August 2021, 60,000 by the end of 2022. Each phase informed the next, adjusting placement, substrate density, monitoring frequency. In the process, Glenmorangie and Harriet Watt University worked with rural Scottish oyster growers and broodstock providers across the country. A government-funded report found that for every aquaculture job created in the Highlands, there was a gross value added of around £68,000. Breeding oysters for reef restoration was adding economic value to some of Scotland's most remote communities. Glenmorangie commissioned a purpose-built workboat, the Dornoch Dancer, built by a local company in nearby Alness. The boat was a signal. This was no longer an experiment. It was infrastructure. The distillery's anaerobic digestion plant had already reduced its biological discharge into the Firth by more than 95%. The oysters were absorbing the remaining fraction. Nature was handling what engineering had reduced but not eliminated. The target remained 4 million oysters across 40 hectares by 2030. A self-sustaining reef requires density and continuity that takes years to establish. The Dornoch Firth is not there yet, but the water is already different from what it was in 2017. The Dornoch Firth is one estuary on the north coast of Scotland, but what Sanderson's team proved there was being watched from far beyond the Highlands. Native oyster populations have declined by approximately 85% globally. The same industrial overfishing that emptied the Dornoch Firth in the late 1800s swept through estuaries across Britain, France, Denmark, Belgium, Sweden, and the Adriatic. What was once the most common reef-forming habitat on shallow European coastlines has been reduced, almost everywhere, to fragments in memory. The science of restoring it barely existed before deep began. When the project's findings were published and its methodology made available, researchers across Europe took notice. The Native Oyster Restoration Alliance now coordinates projects in Belgium, Denmark, France, Sweden, Romania, and across the British Isles, each one working from protocols that DEEP helped establish. Within Scotland alone, new restoration projects are being planned at Plockton, Ardfern, and the Firth of Forth, where native oyster beds once covered an area the size of modern Edinburgh before being fished to extinction in the early 1900s. Sanderson put it plainly: there are very few examples of restoration at scale in the marine environment. DEEP, he believed, would become an exemplar for projects around the world. A single Firth in the Scottish Highlands, 300 canaries in a cage, and a methodology built from scratch had become the closest thing the field had to a blueprint. The work is not finished, and Sanderson does not pretend otherwise. 90,000 oysters is not 4 million. The oyster population is not yet self-sustaining. Larvae still need to be grown in nurseries, transported, and placed by hand. The project depends on continued funding, continued partnerships, and continued political will to maintain the Firth's protected status. And the Dornoch Firth is one Firth. Scotland's coasts once carried native oyster beds across hundreds of kilometers of sheltered sea. Almost all of it is gone. What DEEP has demonstrated in one estuary has not yet been replicated at the scale the broader collapse requires. Shellfish reefs are among the most endangered marine habitats on Earth. That is not a Scottish problem. It is a global one. In March 2017, a team lowered 300 oysters into a Firth that had been without them for over a century. Those oysters opened. They began to filter. They survived the winter and the spring, and the team came back to find the water around them slightly different from the water anywhere else in the Firth. Clearer, quieter in the way that water gets when something living is working inside it. The Dornoch Firth had carried oysters for 10,000 years before the dredgers came. It took less than a generation to empty it. It will take longer than that to fill it again. But the 300 survived. And the Firth, it turned out, had not forgotten them either.